Good afternoon. Welcome everyone to the fourth talk in the webinar series on genetics and evolution organized by the Journal of Genetics. I'm very happy to welcome Professor Stephanie Bedhomme. She works at the Center for Functional and Evolutionary Ecology at Montpellier, France. Stephanie got her PhD from the Genetics and Evolution of Infectious Diseases Laboratory at Montpellier, and she used a mosquito system to look at evolution from a vector's point of view. After that, she did her postdoctoral stint at Queen's University at Kingston, at Münster, Germany, and Valencia and Barcelona, Spain, before moving to her current position. Her research has been quite varied. She has done very interesting stuff. Currently, she works on the consequences of horizontal gene transfer in eukaryotes, specifically from the point of view that insertion of an extraneous gene can break down the co-adaptation between gene and genome. Previously, she has also worked on the evolution and expression of virulence, sexual conflict using a Drosophila system, the effect of historical contingency in viral evolution, and the evolution of generalist-specialist strategies in plant viruses. So I welcome Stephanie, and she will be talking to us today about hypermutator evolution during adaptation to different types of viruses. Stephanie.
Thanks a lot, Prasad. First, I would like to thank the organizers for inviting me to give this webinar. I guess in the pandemic we are going through right now, one of the positive points, because I think it's good to stay positive, is that we got the opportunity to give webinars for all across the world, and probably we realized that we could do that more often. So I will present you experimental data today that are quite new to me. I'm at this point where I was a bit tired of my old talk, and I decided to put up a new one. So I'm very excited about the data, but it might be that I haven't fully digested some of the pieces. So you will see that there are still some areas of small shadows and things that I don't fully understand, but I'm happy to share that with you. We'll start by talking a bit about mutation. So mutation is one of the four main evolutionary forces, and mutations are the raw material on which selection is acting. There are different types of mutations, and I think we quite often only focus on the SNPs, so the one base pair substitutions. They are the ones that are looked at in particular for molecular evolution. But the other types of mutations can have a pretty big role in evolution, and we will see that in the experimental data later. So the other types of mutations are insertions, replications, and deletions. And there are also different origins of mutations. So they can be replication errors. They can be due to external mutagens, like UV, for example. They can be due to transposable elements that will insert within the genome, and these transposable elements travel intra or intergenomically. And there can be intragenomic homologous recombination that is sometimes actually due to the insertion of transposable elements that generate homologous regions in the genome. And we can link the different mutation origins to some of the different mutation types, but not all of them. And something that I think is important to keep in mind is that the mutation rate is not constant over the genome for all mutation types. It's actually variable along the genome. And for some mutation types, the mutation rate is strongly dependent on the local DNA sequence. So for example, some transposable elements will have favorite sequences in which they will insert. So now the mutation rate is not something that is fixed. It's a parameter that can change either plastically or that can also evolve. And the point at which the mutation rate is at a certain moment is actually resulting from the balance between two forces. On one hand, the vast majority of the mutations are deleterious, which is generating a selection pressure to reduce the mutation rate to avoid the deleterious effect of this mutation. And on the other end, the proofreading mechanisms, that is increasing the DNA replication fidelity, and so reducing the mutation rate, these mechanisms are costly. So I just say that the vast majority of the mutations are deleterious. It's true, but it's more or less true depending on the situation. Actually, you have here on this graph the distribution of the fitness effect of mutation that is derived from a Fisher geometrical model, and you have three distributions. These three distributions differ by the distance to the optimum at which a population is. So if the population is at the optimum, it means that the fitness cannot be higher, and any mutation would actually reduce the fitness and have a negative fitness effect. That's the blue curve. But if we go a bit further from the optimum, then the distribution of the mutational effect is getting wider, and you have a proportion of the mutations that then become advantageous. So now you see that the fraction of the mutation effect that is on the right of the zero, that now we see in color, is getting bigger when we go further from the optimum. And being further from the optimum means that, for example, the population is put in a new environment to which it's not adapted. And so this change in the proportion of mutation that can be advantageous is going to change the optimal mutation rate. So the optimal mutation rate is going to be higher if the population is put in a new stressful environment. So now, what are hypermutators? They are genotypes with an increased spontaneous mutation rate compared to a wild-type ancestral genotype. Where do we find them? We find them in different situations. So they have been spotted in populations that have been experimentally evolved in bacteria. So that's the case, for example, in the long-term experimental evolution of the Lenski lab. Now we have also identified a lot of hypermutators in clinical samples, and also quite a lot of cancerous cells are actually hypermutators. Mechanistically, these genotypes are due to the inactivation of one of the methyl mismatch repair system genes. The methyl mismatch repair system is a complex of proteins that is scanning the DNA after DNA replication, is spotting the mismatches that can be generated by replication error, and correcting these mismatches. So if one of the genes of this system is inactivated by a disruptive mutation, or even by a SNP, then this correction or this scanning of the DNA is not occurring, and the mutation rate is strongly increasing. It's actually the rate of SNP and one base pair indel mutations that is increasing, and depending on the gene of the methyl mismatch repair system that is inactivated, a different mutation spectrum is produced. So, for example, some inactivation are going to increase the transition rate, whereas some others are going to increase the transversion rate. So now we can wonder why sometimes hypermutators propagate in the population. Something I forgot to say on the previous slide is that, for example, the hypermutator in the Lenski experiment have a mutation rate that is 100 times higher than the one of the ancestral wild type. So that's quite a strong increase in the mutation rate. So the situation in which hypermutators propagate in population has been described as the following event. So first, the mutation is making one cell hypermutator. This hypermutator is going to generate a lot of mutations, and among them, one is advantageous in the stressful environment where this population is. And because the reproduction of bacteria is mainly clonal, the hypermutator is linked to the advantageous mutation it has generated, and the hypermutator is hitchhiking on this advantageous mutation, and both of them increase in frequency in the population. So there is an initial strong advantage of the hypermutator that is actually the advantage of the advantageous mutation that is generated. But then the hypermutator is also generating a lot of detrimental mutations. And so the advantage of the hypermutator is transient. And the advantage is due to the fact that it's increasing the provision of adaptive mutations. So the hypermutators have been studied for quite a long time. I've put here one of the first experiments by Chow and Cox in '83, where they created mixes of hypermutator and wild type in different proportion, as you can see on here on the y-axis, and they followed the proportion of hypermutator for a bit more than 100 generations. And you can see that above a certain frequency threshold, the hypermutators increase in frequency because, and it's interpreted as they are beneficial because they increase the provision rate of beneficial mutations. So, as I said, hypermutators have been studied for quite a long time with different approaches. So the first one is competition between hypermutator and isogenic wild type. So that's, I've put one of the most recent examples. These experiments always try to see in which conditions hypermutators are more likely to thrive, but they very often start from a 50-50 mix. And if they start for mixes that are have a lower frequency of the hypermutator, there is always a point where the hypermutator is not invading, which is asking the question, how do they invade in natural population? Because they probably actually happen only in one cell. Other approaches are modeling, which are kind of putting into a model the verbal argument I gave you some minutes ago to explain how hypermutators can invade a population. Another approach is what I call the opportunistic detection of hypermutators in experimental evolution. And then people try to characterize the mechanisms that generate them, the fitness advantage that they provide, and how they are maintained in the population. And there is actually very few forward approaches. So people that set up an experiment with the goal to check for the propagation of hypermutators. So this is the example of such an approach, which has been set up in Pseudomonas fluorescens bacteriophage system. So there has been close to 36 co-evolving populations, so of Pseudomonas fluorescens and this bacteriophage. And at the end of the experiment, they checked for the mutation rate of the bacteria. And what they see is that 9 of the co-evolving populations became hypermutator, whereas none of the populations that were evolving alone of the bacteria became hypermutator. And they show that these hypermutator populations are better at eliminating the phage. So that's an example of a stressful environment that is selecting for a hypermutator that is providing an advantage to the population. So now what we have done is one of this forward approach, and we took the advantage of having spotted a hypermutator-prone strain. So this strain is E. coli DH10b, and it's prone to become hypermutator because it carries an insertion sequence stem. So an insertion sequence is the simplest expression of a transposable element. So I've drawn a small graph here. So it contains flanking inverted repeats here in blue and a gene coding for a transposase with its promoter. And that's it. ISN can insert in at least 2 of the method mismatch repair system genes and disrupt it, and so generate a hypermutator. And ISN has the characteristic to have the highest transposition rate ever documented. So it's transposing a lot. So the general design of the experiment we wanted to conduct was to start with this hypermutator-prone ancestor and follow the propagation of hypermutators in different stress levels. So we discussed quite a lot of which stress we were going to use, and we decided to use 2 of them, and for each type of stress to have 3 concentrations of the stressor that correspond to 3 stress levels. And of course, we had control populations that evolved in rich medium, an environment that we consider not to be stressed. So the 2 stresses we have chosen are high salinity on one side and sub-inhibitory concentrations of gentamicin on the other side. So gentamicin is an antibiotic that is targeting the ribosome, whereas the high salinity is likely to affect many functions in the cell. So we chose these 2 stresses because we think that salinity is likely to have a larger mutational target, so a larger number of genes that can potentially be mutated and have a positive effect in the presence of salt than sub-inhibitory concentrations of gentamicin. But that's something we will verify with our experimental data. So the questions we wanted to answer with this experiment is, do hypermutators increase sooner and faster in frequency under more stressful conditions? Can we identify the mutational target in both stresses? Are the mutational targets the same for the wild type and for the hypermutator population? And are these mutational targets different for different doses of the stressor? So the first thing we had to do was to choose the stress levels, and so we did classical dose response curves for gentamicin on the left and for salt on the right, and we chose the concentrations of both stressors that were reducing the population size after 24 hours of growth by 20, 40, and 60%. We actually tried to choose the concentration based on the reduction of growth rate. That would be something that is closer to the fitness, but it revealed to be quite complex, and in particular because in salt the dose response curve is not constant across time. And if you're interested in this, I can tell you after, or ask me the question after the talk. So now we have our experimental plan. So we evolved population of bacteria for about 1,000 generations with these stressors. We had a daily transfer with a 1 to 100 deletion. We had a replication of 12 for the 6 different concentrations of stressor and 24 for the LB population. So I will mainly present genetic data today, and so the 2 big things we did in terms of checking what's happening at the genetic level is to detect the insertion of the IS10 in the mut-S and mut-L gene along evolution, thanks to a PCR test. And then we acquired the whole sequences of the genomes at the population level after 1,000 generations. So I said I will mainly present genetic data, but I can't resist showing you some phenotypic data for the populations that evolved in gentamicin. So what you see on this small graph is the minimum inhibitory concentrations of the populations after 1,000 generations of evolution. And so here on the left, you have the populations that evolved in LD. You see that they all have a minimum inhibitory concentration of 16 micrograms per milliliter. But then you have populations evolved in 0.5, 1, and 2 micrograms per milliliter of gentamicin. And you see that first, they present a diversity of MIC. So a single condition of evolution is not always leading to exactly the same phenotype. And also that depending on the concentration of selection, of evolution, we have a different pattern of MIC. So the higher the concentration of evolution, the higher the MIC of the evolved population. If we put that in another form, along this concentration arrow, you have here the concentrations at which the population evolved. Here, the minimum inhibitory concentration of the ancestral population. Here in black, you have the MIC of the populations that evolved in LD. And now you have here the MIC of the populations that evolved in the higher concentration of gentamicin, but concentrations that are still very low since they are here. So I think it's quite interesting to see that there is a strong difference between the concentrations at which they evolve and the MIC that is selected. And in particular, that concentrations that are really sub-inhibitory can select for MIC that are at the level or above the target plasmatic concentration of gentamicin when it's used in clinic. So now, going back to the hypermutator story and the genetic determinants, here I show the result of this PCR test that we did every 100 generations. And you have, so each horizontal bar represents a population across time. And in red and blue, you have populations where we detected an IS insertion within the mut-S gene in red and within the mut-L gene in blue. And when the bars are hatched, it's because we could detect the presence of bacteria that had the insertion mixed with bacteria that were wild type. So what we can see on this graph is that, so here on the top you have populations that evolved in gentamicin and then populations that evolved in salt at the bottom. I didn't represent the LD population because none of them got an IS insertion within one of the mut genes. What we can see here is that IS-triggered hypermutators propagate only in stressful environments and much more so in salt than in gentamicin. And something that I also find quite interesting is that we have long periods of coexistence of the hypermutator and the wild type. So that can run for more than 500 generations. So this means that this model of advantageous mutation on which the hypermutator is hitchhiking is probably true, but it's not leading to the fixation of the hypermutator very rapidly. And I think this can be due to the generation of a lot of other mutations and very strong clonal interference going on within this population, this hypermutator population. So now we have acquired the full genome of the 96 evolved population. So here, what you see now on the screen is the same information as on the previous slide, but only for generation 1000. So that's in blue populations that have an IS insertion in mut-L, in red populations that have an IS insertion in mut-S. And now if I add the information that we got from the whole genome sequencing, there are additional hypermutators that we hadn't detected before. These additional mutators are due either to mutations in other genes of the methylene mismatch repair system, so namely mut-P, DNA-Q, and mut-H, or SNP or small indels in mut-S and mut-L that we couldn't detect by a PCR test. So we have detected these mutations that we know are very likely to generate hypermutated genotypes. But we have also checked the number of mutations that were accumulated in this population, and this is confirming that we have hypermutators. We have also measured the mutation rate for some of them by classical Urea-Delbert protocols. And so here I'm just showing you the average number. So the number of mutations, or non-IS mutations per population at generation 1000 for the populations that stay wild type, and here in pink for the populations that became hypermutated. So you see that we have a big spread that is probably due to the combination of time since hypermutator appearance, because here we are at generation 1000, some of them became hypermutator at generation 200 and some other 800. And also that depending on the gene on which they are mutated, they might have different mutation rates. So if we ask one of our first questions, so do conditions of evolution influence the propagation of hypermutators? The answer is yes. So we have a significant difference between these three conditions for the number of populations that became hypermutator. And it's actually salt that is different from LD and gentamicin. But then if we go a bit deeper in the result and we split these hypermutator populations in non-IS triggered hypermutators and IS triggered hypermutators, we see that this pattern is actually due to the fact that there are a lot of IS triggered hypermutator in salt and only one population, I think, in gentamicin. And then when we saw that, we wondered whether it could be due to a different IS transposition rate in salt. So we actually compiled all the IS10 insertion that we could find in all the datasets, so not only in the MMRS genes, but across all genomes, and that's what we obtained. So that's the number of new IS10 insertion that we could find across all the dataset for populations evolved in gentamicin, for populations evolved in LD, and for populations evolved in salt. So you see that we have a very strong, very strongly significant increase of the number of new IS10 insertions in salt. So salt population have more IS10 insertions. So now we wonder why. So it could be that salt is increasing the competency, the natural competency of the cell, and that cells are transformed by IS10 sequences from the extracellular environment. It could be that IS10-induced mutations are advantageous in salt, and so that they are selected for, and that's why we see more. Or it could be that the IS10 transposition is actually increased or triggered by salt or by a stress response that is itself triggered by salt. So we try to discriminate between these different hypotheses. And for this, we looked at the other IS insertions. So for now, I focused only on the IS10, but there are other IS in this genome. And there were also new insertions of this other IS. So that's here a small table with the different other insertion sequences, the number of new insertions that we could detect in the dataset, and now how they are split between the different environments. So you see that for three of them, we have significant difference between the three environments, but it's not always going in the same direction. So for example, for IS 150, no salt population has a new IS insertion. As for IS 186, that's the contrary, the salt population has more insertion. So the environmental effect seems to be IS specific, which is kind of an argument against a general increase in competency. So if it was a general increase in competency in salt, we would expect to have the number of new insertion of any IS to be higher in salt than in other environments. So then we tried to see whether we had indices that IS insertions were advantageous in salt. And so what we did was to check the number of new IS insertions in the mutational target. So I will come back soon on how we define the mutational target. But here you have the number of new IS insertions in the mutational target for gentamicin and for salt. So we actually have a higher number in salt than in gentamicin, but we have on average 2.9 extra IS10 insertions in the salt mutational target compared to gentamicin. And if we look at the general data in the whole genome, the difference is of about 17 additional IS10 insertions. So selection might actually play a role in this increase in the number of new IS insertions that we see, but it cannot explain all the difference. So if we sum up, it doesn't seem to be an increase in the competency that is due to salt. Selection can explain part of it, but does not explain all the extra IS insertion that we see in the salt population. So our data points to a higher IS10 transposition rate in presence of salt. That could be due to an induction of the transposase promoter by salt or by stress response. So now what are the consequences of this, this seemingly higher IS10 transposition rate in salt population? it means that soil populations have a higher provision of ISM-triggered hypermutators. So we've evaluated that it's about 3.5 times more from the data we have. What we also see is that there are other ways of becoming hypermutators, and that actually gentamicin populations are using them. So we saw more of these other ways in gentamicin populations. And so the higher number of hypermutators in salt is thus probably partly due to a higher hypermutator provision. And with the replication level we have, it's very difficult to test whether it's only due to that or whether there is actually really a strong higher advantage for hypermutators in the salt population. Something that I think is interesting in this story, even though it was, as you can imagine, a bit, we actually didn't expect all the ISM transposition rate to be variable across environments. We have the salt population that kind of become first hypermutator for the ISM transposition rate. So they have the rate of ISM transposition that is increasing. It seems to be environmentally triggered. So we actually don't find any evolution of the transposase sequence, for example. And then because of this ISM transposition rate that is increasing, there is a higher provision of hypermutators. That this time it's a genetic increase of the mutation rate. So now something we wanted to do was to define the mutational targets, verify that this prediction of a bigger larger mutational target in salt than in the gentamicin was true, and see how this can be linked to the propagation of hypermutator in the population. So from the sequencing data, we actually compiled a matrix of the genes that were mutated in each of the populations. And so the first general thing that we can see, so out of the 92 populations for which we had good sequence data, there were more than 2,500 genes that were affected by mutations. It means that because we had a lot of hypermutators across the whole data set, more than half of the genes of E. coli were affected. And here I've plotted the distribution of the number of populations for which a gene is mutated. So you have genes, for example, this one, that is mutated in 85 out of 92 populations. So these genes that are mutated in a lot of populations are very likely mutations that increase the adaptation to culture conditions. So that's not the one we're interested in, but it's still interesting to see that they exist and that there is a very strong convergence of these genes, on the evolution of these genes. So what we want to know is genes that are specifically mutated in certain evolution treatments. And we derive from this mutation in parallel that these mutations are likely to be adaptive in this situation. So to determine this, what we did is to calculate the number of genes that is mutated per population, and from there to construct a null model where the hypothesis was that mutations occur randomly across all the genes. So from this probability of a gene to be mutated in each population, we could derive for each population group the number n such that the probability that the number of populations in which a gene is mutated is superior or equal to n, such that this probability is below 0.01. So it's looking for a threshold in terms of the number of populations in which a gene is mutated, for which the parallel mutation is not by chance. So it's likely to be not by chance. So we obtained a list of genes that was mutated in parallel for each group of populations. So by group of populations, I mean a group of populations with the same stressor or with the same concentration of stressor. And the mutational target in group A was defined as the genes that were mutated in parallel in group A minus the genes that were mutated in parallel in LD, which is our control. And so we expect that the genes mutated in parallel in LD are genes that increase the adaptation to the medium and to the transfer protein. So doing this, we find a mutational target in gentamicin of 35 genes and of 69 genes in salt, with 6 genes in common. So we kind of verify our predictions that the mutational target was going to be bigger in salt than in gentamicin. Then we can apply this definition to each of the concentration of the stressors. So here you have the mutational targets that are defined on each of the concentration of gentamicin and the intersections of these mutational targets. So I put the whole list of genes. I'm not going to talk about them separately. Just to give you a picture of what we get. So we have a lot of genes that are only in one of the mutational targets, so in the mutational target for one of the doses. And this kind of suggests that the mutational target is composed of a small set of genes that is mutated in the 3 concentrations and many genes that are only mutated in one of the concentrations. So part of the genes that are here in the intersections, so the intersections between the 3 and also this one and this one, are genes that had been identified before as potentially playing a role in aminoglycoside resistance. And it's also interesting to see that we have 6 genes of the NEO operon that are in the different mutational points. I will come back later to that. So that's conducting the same analysis on the salt mutational targets for the 3 concentrations. We have a pattern that is quite similar, and also here 2 operon that seems to be mutated in different concentrations. Another way of defining the mutational target could be to look at the union of the 3 mutational targets for individual concentrations. If we do that, we have again a picture of a larger mutational target in salt than in gentamicin and a quite small intersection between the 2 mutational targets. So I just want to share with you some thoughts about this definition of the mutational target. So I present that quite fast, but we actually had quite a lot of discussions on how to analyze and define this mutational target. And something that I think is quite important is that some of the genes that we find mutated in parallel could actually be genes that carry compensatory mutations. It means that because we look at the set of genes that is mutated after 1,000 generations of evolution, population had time to have first mutations that improve the fitness in presence of the stressor, and then mutations that reduce the fitness cost of this initial mutation. So I think for now we cannot really distinguish them. We could look at specific associations or disassociation between individual mutations, although our replication level is a bit small to do that. But we won't have information on the order in which this mutation occurs, which is necessary to really separate the compensatory from the initial ones. And then I'm asking, is it a problem? Because actually, if there are more compensatory mutations, these compensatory mutations actually have a beneficial effect. And so in terms of selecting for the propagation of hypermutators, they probably have a similar role as the initial mutation. So in the context of looking at a link between the mutational target and the hypermutator propagation, I don't think these compensatory mutations are a big problem in our reasoning. And the second part is kind of questioning the use of parallel evolution to define the mutational target. Because actually, if you think about it, the broader a mutational target is defined. So this time defining the mutational target as all the potential mutations that are improving the fitness in a new environment, then it's less likely to have populations mutated in parallel. Actually, if you go to the extreme, if we have as many evolutionary paths as replicate population, each of them could follow a different path. They would adapt to the mutation target would be, but we won't have any parallel evolution. So that's actually like a theoretical extreme case. But I think it's good to keep that in mind, and I actually have no way of solving this question, but I just wanted to share this reflection with you. So now, quickly, before I finish, I wanted to show you some slides on the nature of the mutations in the mutational target. So these two graphs have nothing to do with American elections. There are the proportion of non-disruptive and disruptive mutations for each of the genes that we found to be included in the mutational target. And so you see that for both mutational targets, we have a series of genes in which all the mutations are non-disruptive. And then we have genes, and there are actually quite a high proportion of the mutations, for which the proportion of disruptive mutation is quite high, and some of them, all the mutations are actually disruptive. So that's the case also here. So you see that this, for example, here in salt, these two genes that are quite heavily mutated are very majority mutated by disruptive mutations. So it's adapting by breaking a gene. So now if we focus on some of the genes, so fuse A, which is one of the genes that is heavily mutated in gentamicin, it's encoding an elongation factor, and it's catalyzing the coordinated movement of the two transfer molecules, the messenger RNA, and the conformational changes of the ribosome. And it's thought that the mutation in fuse A are reducing the binding of the antibiotic to the ribosome. So if we look within fuse A at the level of parallel evolution, we had 20 populations that were mutated in this gene. 25 different mutational spots have been identified. 25 mutations have been identified. 12 positions are mutated, and 7 of these positions have a unique mutation. But so you see that some spots are heavily mutated in parallel. So there is some level of parallelism within the gene. And also you can see that all the mutations are in the second half of the gene and concentrated here around this point. Another example I wanted to show you is the NUO operon. So the NUO operon is encoding the 14 subunits of the proton translocated NADH ubiquinone oxidoreductase. It's actually proteins that are involved in the electron transport chain in the membrane of the bacteria. It's known that the aminoglycoside antibiotics are activating the stress response, the envelope stress response, and that the expression of the respiratory envelope is toxic when the stress response is activated. So that's probably why breaking these genes, so breaking these respiratory proteins, is increasing the fitness in presence of gentamicin, and actually breaking any of the subunits seems to play the same role. So, well, that's actually the location of the mutation in the operon, but I think I prefer to show you this. So, is there an effect of being hypermutator on the mutational target? So here I've plotted the number of mutations in which a gene is mutated in the wild-type population and in the hypermutator population on the y-axis. So that's for genes of the gentamicin mutational target. And here I just remind you that 6 out of 75 populations are hypermutated. And you can see that there are 2 genes that are mutated only in hypermutators, you know, the one in red here. And yeah, the size of the dot is proportional to the number of genes. And here we have 5 genes that are mutated only in the wild type. So here we don't have a very strong effect of being a hypermutator or not on the mutational target. But if we go to salt, we actually have 18 genes here that are mutated only in the hypermutators. And for now, I don't quite understand why this is. So the mutations are mainly SNPs, and I don't see how they are different from the SNPs that could be acquired in the wild type population. But I think it's still interesting to see that these 18 genes out of a mutational target of 69 are only mutated in the hypermutator. So it seems that in SALT, being a hypermutator is opening access to new parts of the mutational target. So to conclude, we wanted to know the hypermutators increase sooner and faster under more stressful conditions. I think the answer we gave is yes, but part of it is mediated by a higher transposition rate of ISN at high salinity, so the specific mechanism in our experimental design. Can we identify the mutational target in both stresses? We have identified a mutational target. I'm still thinking about this mutational target definition. But with this definition, we see a difference in the mutational target size, and it could be linked to the hypermutator selection. But we need, I think, here to adopt a theoretical modeling to really establish this link between the two. And in the mutational target, some of the genes were previously identified to provide resistance and tolerance to aminoglycoside and salt, which is comforting us in the right way of defining the mutational target. So is the mutational target the same for wild type and hypermutator population? In SALT populations, we seem to have hypermutator specific mutations. And finally, are the mutational targets different for different doses? So here we seem to have a structure of a mutational target that is composed of a small set of genes that is mutated in the 3 doses, and then many genes that are mutated only in one of the doses. And so what we are doing now is that we have isolated hypermutator and wild type clones from right at the beginning, so when they start to rise in frequency in the population. We are sequencing this clone and doing competitive fitness measurements to really go to what is the advantageous mutation on which the hypermutator is exciting, and what is the fitness advantage that this mutation is providing, such that there can be an increase in frequency in the population. And we are also trying to follow the dynamics of the propagation of the hypermutators, because we were quite surprised by this long coexistence of the hypermutator and the wild. And finally, this project was really a common project of all the members of the lab. So some of them are not here anymore, so there has been mandatory participation to the project. So Caroline Rose, the postdoc who really designed the experiment and did a lot of the follow-up. Mike Finnegan is technical assistant. Lena was a master. And Martin is also a postdoc. Leah, PhD student. And the genomic analysis were mainly done by myself. I also would like to thank the ERC and the CNRS for funding, and you for your attention, and I'm very happy to answer your questions.
Thank you, Stephanie, for a very nice talk. I already see that there are a large number of questions, so maybe we can start taking them. First question, it's actually related, 2 questions related. Do you have any idea why so many more genes mutate in one specific gentamicin concentration compared to in all 3 gentamicin concentrations? And related to that is, what kinds of mechanisms in terms of gene function might be involved in generating this pattern?
So maybe I can go back to the... So I think it's related to this graph. So we have many more genes that seem to be mutated in so G1 and G2 than in G3. I don't really have a clear explanation for why this is. So actually, we had no G3 population that became hypermutator. So in general, these populations have less mutation. But this should be taken into account in the null model. So Then for the function of the gene, so I showed you that fuse A was an elongation factor. Another gene that is heavily mutated is TRKH, which is actually a proton transporter, so transmembrane, so it's thought to be involved in establishing the proton gradient against which gentamicin is entering the cell. So by modifying this proton gradient, it's probably preventing gentamicin to enter the cell. So something that we have to keep in mind, and that might actually be a small problem in this mutational target definition, is that it's very likely, it's possible that a gene that is only in G1 is actually also mutated here, but in only one or two populations, such that it's below the threshold defined for this concentration. So I probably would have to find another representation, because it's true that still we get the impression that these genes are only mutated in G1 populations, that they are more mutated in G1 populations than in the other.
Okay. The second question is, what is the relative abundance of mutations that are the same across replicate populations within selection treatment versus those differing among replicates?
I don't have the numbers in mind. So by identical mutation, it depends. We can define that as exact identical mutation, so same SNP in the same position, or we can define that in terms of the same gene mutated. And so for the second definition, I don't really have the numbers. What I can show you is here. So as you can see here, so that's for the whole data set. So the control, the salt, and the gentamicin population. And you can see that more than 1,000 genes are mutated in only one population. So there are, I guess, what you call individual mutations. And so if that's 1,000, I would say that more than 500 are mutated in only two populations. So that's the distribution of the number of populations in which a gene is mutated. So we have 2,568 genes that are affected by a mutation, and among them, 1,176 are mutated in only one population. So that's kind of an answer to the question. Then we can also check this at the gene level. So that's what I showed on the fuse A gene. And here I don't have in mind the result for all the genes that are highly mutated, but you can see again that within a gene, in terms of exact position that is mutated, we also have a pattern of 7 out of the 12 positions that are unique mutations, and then 5 positions that are repeatedly mutated across populations.
Okay. The next question is from Putul Banerjee, and the question is there a correlation between the distance between the original transposon site and the methylation mismatch repair genes in the rate of hypermutator generation?
I haven't thought about that, to be very honest. So I didn't check it. So there is initially, so in the ancestral genome, there are 3 copies of the ISN. So I could check the distance of these 3 copies to the methyl mismatch repair system genes. The methyl mismatch repair system genes, I actually don't know if they are all together in the genome. But something that is a bit difficult to answer is that during the evolution, there are going to be new copies that are going to happen in the genome that are not necessarily in the mute genes. So I could check and I could answer this question for the ancestral genome, but then the situation is going to change and to evolve across along the experimental evolution. So I guess the idea behind that was that it could be that this strain is very prone to become hypermutator because it carries an IS10 that is close to the methylene mismatch repair system gene.
Okay. One more question from Nirmal, who asks, which are the other major hypermutators than the one that you have studied?
Well, I think they are really the main ones. So there are other genes of the methylene mismatch repair system that can be mutated and trigger hypermutators that we didn't find in our dataset. So that's the case of UVR A and UVRB, I think. But there is a single system of so to check of proofreading after the DNA replication. And I know that in cancer cells, the hypermutators are also due to mutations in the equivalent of the CNRS. So that's a system that is very conserved. So that there are you can find equivalence between the system that in bacteria and in eukaryotic cells.
So a related question is, are hypermutators known in eukaryotic system?
Yes, cancerous cells are hypermutators. So in eukaryotic systems, so I actually don't know about examples. Yeah, there are hypermutators in yeast. So I was looking for an example of unicellular organisms. In yeast, there are hypermutators. And then the one that are most described are the one of cancerous cells. But here the implications are a bit different because you have a clonal propagation of these hypermutator cells that have like a local fitness advantage. But of course, the effect for the full organism is very different. So here you have a kind of like an evolutionary conflict within this multicellular organization.
Okay. I guess I have one question, which is you've done this beautiful long-term experimental evolution studies in bacteria for this purpose. Would you like to speculate how things might change or affect if the environments that you used were a little bit more complex or maybe varying over time? For example, if you had a combination of salt and gentamicin or concentrations that varied over time, especially with respect to the mutational targets that one is looking at.
Well, I don't have clear prediction of what would happen, but I guess if we have conditions that are varying, it means that the advantage that is provided by the advantageous mutation that then is like taking the hypermutator with it is going to be to vary across time.
Yeah.
So I would kind of predict that there would be even longer coexistence between the hypermutator and the wild type, and maybe also even more cases where the hypermutator can initially increase in frequency in the population, but then disappear. So we actually had one of these cases, but I didn't point in our data. But in other cases, each time we had a hypermutator that started to rise in frequency, it either stayed in combination with the wild type or got fixed at further generations. But I guess if we are in a more fluctuating environment, we would have more of this transient increase in frequency of the hypermutators and then decrease. That's something that I think is very difficult to predict and even to study, is the crazy clonal interference that is likely to happen within the fraction of the population that is hypermutator. And that's going to increase a lot of like almost stochastic behavior of the population.
Okay. I guess we have answered pretty much all the questions. So I would like to thank Stephanie for a fantastic talk and for joining us today. And I would like to thank all of you for participating. Thank you so much. Thank you, Stephanie.